Hello, in this chapter we will be focusing on live dyes. For some applications, it is impossible to generate transgenic lines to carry out live imaging. As such, several live dyes have been developed to enable imaging of cells that do not carry fluorescent transgenes. In this section, I will explain first the concept of live dyes, then how a principal staining could be achieved to then introduce the most common application techniques. In the end, I will summarize the advantages and disadvantages of using live dyes. Live dyes are generally small proteins or peptides that specifically bind to an epitope of the protein or structure of interest, resulting in its labeling. They can also be tagged to a protein which changes conformation in response to cellular signaling, thereby activating the fluorophore. Importantly, instead of antibodies, live dyes can also be developed to intercalate into specific membranes with a fluorophore tagged to a specific lipid, and thus not only provide information on protein but also organelle localization. In general, a live eukaryotic cell is cultured in the presence of a live dye, which is transported into the cell to then bind specifically to the structures of interest, in our case the mitochondria. Such a staining would allow live imaging of mitochondrial localization during cell migration, for example. Live dyes are used in two major fields, not taking into account all photoconvertible dyes used for super-resolution techniques. They can function to yield information of the cellular state. As such, ion sensors have been converted into live dyes, resulting in the possibility to perform calcium imaging and thereby gain insight on neuronal activity of live brain tissue, or calcium signaling during embryonic development. To assess the levels of apoptosis upon specific processes, for example during development of a mutant, dyes that detect cleaved caspase 3 activity have been shown to be useful. Through these we can follow cell death within tissues. Another field where live dyes are employed is cell tracking. For this, we can either use lipids, which intercalate with the cell membrane to allow the reconstruction of single cell shapes, or nuclear dyes that are more favorable to use for common tracking softwares and can provide information on mitotic status of the cell image. Live dyes are common tools and bear many advantages. First of all, an experiment using live dyes can be carried out without much preparation, as there is no need for any genetic alteration in the organism of interest. But at the same time, live dyes have strong time limitations due to photobleaching, since there is no possibility to add further dye during one experiment, whereas transgenic organisms continuously produce the protein of interest. Several dyes can only be used in very specific media, as they exhibit strong background otherwise. This places a lot of limitation, especially for mammalian cell or embryo culture, as the media used there tend to be very rich in supplements, which prohibits the proper localization of the dyes.